Welcome everyone to the Evolution Revolution Summit by PNI. This session 2 is about partnering to build the genetic medicines of the future. And we have 3 great panel members here who come from different therapeutic areas, but are bound by a common thread of working and using mRNA LNPs. And Precision Nanosystems collaborates, partners with all these 3 companies and more. We have invited these 3 who are able to provide a perspective into the commonalities and differences within the areas that they work in and just what are the critical aspects. So I would like to invite Professor Robin Shattuck from the Imperial College of London, Sharon Wong, Vice President Technical Development at Ultragenyx Pharmaceuticals, and Kurt Kamruth, Director of Genomic Platforms at Ceva Animal Health. So if we can get yourself to unmute and be on video, we can then get started. Okay, I think I see Kurt, Sharon, and Robin. Perfect. Jane, is it looking fine? I mean, are we good with the broadcast to get started?
You're perfect. We're ready to go.
Okay, thank you. Thanks, Jane. So Robin, for each one of you, and we'll start with Robin first. Thank you for joining this panel session here today. I'm excited about this. So maybe Robin, we can start with you. If you could introduce yourself, the work you do, and also in the context of the companies, 2 companies that you have more recently founded.
Hello to everybody. Yes, I'm Professor Robin Shattuck. My day job is at Imperial College London, where I'm a professor in mucosal immunity. But we have been developing a self-amplifying RNA platform for pandemic use. That was initially funded by CEPI. It's still funded by CEPI in terms of developing the platform for Disease X. But in the context of COVID-19, we've been developing a self-amplifying RNA against COVID-19, which is now in late phase 2 development. And at the beginning of this year, we set up 2 companies, which are kind of sister companies. One is called VaxEquity Global Health. That's specifically dedicated to developing a COVID-19 vaccine. And it's run on social business principles in that it's dedicated to making the vaccine available at affordable cost on a global basis. And its sister company, VaxEquity, is a fully fledged commercial company, which is going after commercial infectious disease targets. That's where we are right now. Very encouraged by the recent data from Pfizer, BioNTech, and from Moderna.
Thank you. Sharon, if you could introduce yourself and the work you do in the company.
Yeah, thank you, Padma. Hello, everyone. I'm Sharon Wong from Ultragenyx. I lead the technical development function for Ultragenyx. Maybe just a little bit about Ultragenyx. We're a biopharmaceutical company that's focused on the development and commercialization of novel products for rare and ultra rare disease. And we have a fairly diverse product portfolio covering commercial products as well as pipelines focusing on. We focus on high unmet medical need, clear biology with no approved therapies. We do have multiple modalities in our pipeline. We work on typical biologics, proteins and antibodies. We work on small molecules. We also work on gene therapy and messenger RNA products. So in the LNP RNA space, we have a collaboration with Arcturus Therapeutics. They are a messenger RNA medicines company. Under that agreement, we have the ability to use their technology to develop products for up to 12 rare disease targets. I think that's all I want to say. Thank you.
Great, thank you. Kurt Kamrud, if you could talk a little bit about, again, yourself and SIVA Animal Health.
Great, yeah, thank you. As Padma said, my name is Kurt Kamrud. I'm the director of the Genomic Platforms program here at SIVA Animal Health. I've been working with self-amplifying messenger RNAs for most of my career. Early on at a company called AlphaVax using a particle-based approach, and now more recently with the synthetic RNA vaccine approach, which is very similar to what Robin is describing. So I appreciate that tremendously. So SIVA, a bit about SIVA, it's a production animal vaccine company. We focus primarily on poultry, swine, and ruminants. It's an interesting company in that it's pretty forward-looking. It has multiple recombinant live vaccine products, which makes it sort of unique within the veterinary world. We have had an interest in platform systems for quite some time. And when I joined the company, we were able to move in with a SAM system here. This is a very flexible system, as we all know, and we think that as we talk about the economics of the vaccines, it actually has a way forward in veterinary health as well. So thank you for that introduction.
Okay. Thank you. And it's great, Precision Nanosystems, as you know, we have our manufacturing, the NanoAssembler manufacturing platform, which are used by all our 3 panel members and another 200 academic and industry experts all over the world. Also, our library of novel lipids is ready for partnering, and we have some of our panel members here evaluating and using those. We're excited about the collaborations ongoing, with Imperial College London and SIVA. And of course, the expertise from Precision Nanosystems in formulation development, in the scale up. And I know we are partnering with all 3 of you as it pertains to, our formulations team, our scale up and process development teams. So going to the second question, I'd like to understand a little bit more, and maybe Kurt, we can start with you. In the uniqueness of the business model, and as you think about mRNA LNPs for animal health, and you in-license these different components from different partners, what becomes important for your business model, for you to develop and launch products?
That's a great question, and animal health is a really great place to build this sort of this type of technology. We have a couple things that we need to deal with on a global, and then uniquely on a farm-specific or system-specific approach. So globally, of course, we need high volume, so we need scalability and for both the delivery technology as well as RNA. And we've talked a bit about that in the earlier session. But very importantly, for animal health, or at least for production animal health, is the cost needs to be low. So we are looking at two big areas, scalability and low cost from a global perspective. What's interesting and unique currently in the United States is that the USDA has approved what they call a platform approach to registration of vaccines. And if you obtain a full license for a platform system, you can then rapidly use it to do farm-specific or outbreak-specific responses to veterinary concerns. So in that instance, you may not need large volumes, but you certainly need to maintain the low cost. So there's those 2 things are working, sort of together as we build our business model. The guidance, as I mentioned, in the United States for registration is pretty straightforward and has been in place now for a number of years. The EU is rapidly moving in that direction, and we look forward to some guidance that's going to be released in 2022, which will expand this business opportunity into the European Union, and then further as most countries will accept USDA or EU approvals. So I think those are sort of the interesting things that animal health needs. These, of course, are animals that live on the order of weeks or months as opposed to decades. And so the registration process is different than on the human side, and I think that from a business perspective, that's really a huge advantage, and we can look at efficacy very quickly, and as we commercialize products.
Perfect, thank you. Sharon, from the rare disease area, as you license and build products, what do you see as the business model that works best for Ultragenyx?
Yeah, for Ultragenyx, our business model is that we focus on the products to treat rare disease patients. So we don't do discovery research. We also don't have a singular focus on a specific technology. So what we do is we collaborate with and partner with academic institutions, with other companies, identifying the right target for a disease. And then we bring in the right technology to enable development of the therapeutics for the patients. So in that basis, we have a collaboration with Arcturus to bring in the mRNA technology. We have collaboration with you guys, ADMA with PNI, to enable generation of high quality lipid nanoparticles.
Yeah, great. Robin, from, again, the two companies initiated as you look to, get the drugs, the vaccines worldwide, what business models, are you looking at that would work?
Yeah, I mean, I think the infectious disease vaccine space is quite complicated, and, fantastic success with those first two RNA candidates that have shown efficacy against COVID-19. But they're not cheap vaccines, and they're not easy to distribute. And if you look globally, most vaccines that are being used at large scale around the world actually require being at less than $1 a dose, and with reduced reliance on a cold chain. So, I think it's going to be a very transformative period for infectious diseases and mRNA. We do need to bring the costs down. We do need to improve the cold chain in order to make vaccines that are viable on a global basis. And I think to some extent, we suffer, but also celebrate the fact that most of the investment in mRNA-based vaccines has come out of the oncology and personal medicine space, where cost and volume don't tend to be issues. But for this technology to become mainstream in the vaccine field, both of those need to be solved, as does stability. You need commercial success, but I'm also, have a very strong interest in global access to vaccines and technology. And I think that there is the potential for this type of technology to make vaccine production in different parts of the world more viable against commercial targets that might not be globally viable. But if you have small foot production base, then you may actually have a commercial model that works for some of the rarer infectious diseases, or those that are geographically restricted. So I think we're just at a tipping point, beginning of a story. It doesn't completely answer your question as to what makes it commercially success, but we recognize the challenges that are out there and what needs to be solved to change mRNA from being a high income, small volume business to being something that's transformative for global infectious diseases.
Yeah, no, that's good. I mean, you did answer the question with respect to, what is required and some of the challenges. And PNI certainly, has developed a manufacturing platform that is small footprint that can be moved, anywhere in the world for more of a localized manufacturing. Can you speak maybe, Robin, a little bit into maybe more technical challenges? There is obviously multiple components. There is the RNA, there is the lipid nanoparticles, sourcing, there is manufacturability, and then there is, the whole development, you talked about cold chain and, room temperature. So as where do you see as the big bigger challenges, as we want to move these vaccines all over the world, develop them quickly and make them available, in a worldwide space?
So obviously we're working on self-amplifying RNA, and that's a lot larger than regular RNA. So I was quite smiling to myself when Peter Cullis was talking about going from siRNA to mRNA and it being a challenge. For self-amplifying RNA, that's an additional challenge, but it has the advantage of potentially allowing you to go with very low dose, which again, for a global product may be particularly important. The lower we can drive the dose that's required, the more doses of vaccine we have to be able to move around the world. I think that the bigger the volume, the more potential for driving down the costs of the individual reagents. I suspect there will be a challenge, a supply chain challenge for lipids, particularly given the recent successes and a scramble to produce some of these vaccines. I anticipate that will be a short-term challenge. I hope it will actually increase the capacity for manufacturing many of these lipids so that it becomes less of a bottleneck in the future. But that will be important for us and for others. And then the big thing is the cold chain, the ability to have a more stable product that can be shipped more easily. We have to remember that actually it's that last mile in the distribution process that's most important. Getting the vaccine out to remote areas. We've been recently discussing doing clinical trials in Peru, and they're saying, well, how do we get this vaccine out to communities up in the Andes? We can't carry a -70 freezer on the back of a mule to get it there. How are you going to solve that problem for us? So those are the kind of issues that we're looking at and the challenges that we face right now.
Sharon, in your space, challenges as you see as it relate to, again, licensing, partnering, or just technically what you see are the issues as you develop and launch your products.
Yeah, maybe one challenge I would mention is that the complexity of the supply chain. We essentially work with, for each step, we work with a separate vendor. Right? We work with a contract manufacturing organization to make the plasmid, and then another one to make the mRNA drug substance, one to make the lipid, and one to make the lipid nanoparticle and the final fill and finish, as well as the labeling, packaging, distribution, right? And on top of that, we also work with a number of analytical organizations to do analytical characterization of the molecule, as well as testing and release of the product. So the challenge for us is really the resources and the effort that's involved in kind of orchestrating all of these different activities. Timeline is a bit of a challenge as well to work through the make, the test, and the release process for each of these steps before you can move to the next step.
Yeah, Kurt, can you chime in? Is it any easier in the animal world? Or, I mean, again, you have come from synthetic genomics, building human vaccines, then now into the animal vaccines, or you see similar challenges?
Yeah, I see some other challenges from the veterinary, the production veterinary animal health. The economics of veterinary vaccines is quite different than human vaccines, and so when partnering or looking for licensing, we need to communicate that to the potential partner or license company. And for example, so for a blockbuster vaccine in the veterinary world is $50 million a year. So the scale of economics is quite different. So I think that's a challenge. And when we've been successful, we recognize we bring some other value to the table other than just pure dollars. From a production perspective, SIVA is planning to basically make their own RNA. We'll do that internally. We will be partnering for the delivery technology, the LNPs, and the process of putting them together, as those are not currently strengths within the organization we currently have. We are really focusing, at least in the United States. Now, globally, it's going to be different, but in the US, there's, because of this ability to use a platform system to respond to what's taking place specifically in different regions or even specific farms, this just-in-time manufacture sort of idea, and the speed with which you can do that will allow some of the cold chain issues that are clearly an issue when you talk about global to be less of an issue for some of the production animal activities that we look forward to doing. So the temperature component is a big deal, especially as we think globally. At least in terms of the prescription, that's the actual formal term that USDA uses when you have a platform registration to go in and get specific. I think we can do just-in-time manufacture, and some of the stability issues will fall away in that front.
That's good. No, thank you. I'll just do a shout out to our audience. If you have any questions, please do, send them through the Q&A, little box in the down bottom if you have. And again, we will find time. We will make time to make sure we answer. Our panelists will answer those questions. So moving on to the next theme that I want to talk about is, again, mRNA LNPs, have been in research, for many years, as Dr. Peter Cullis and Jeffrey Elmers and Norbert have talked about. But with the whole COVID pandemic, they have, I believe, come really to the front and center stage. The mRNA LNPs, fantastic news coming from Pfizer and, BioNTech and CureVac and others. My question is, and maybe we can start with Sharon, because I know Ultragenyx works with antibodies, with viral vectors, and many technologies, including mRNA LNP. What is going to take for the mRNA LNPs to get to mainstream, right? So this is a new field that is coming fast, showing great data, but what is it going to take for this technology to get to mainstream? Sharon.
Yeah, thank you, Pam, for the question. Maybe I'll talk about a few areas. I would say the first one is to really mature the manufacturing process. The manufacturing process for mRNA and lipid nanoparticle is still at fairly early stages relative to what we're able to do for antibodies, for example, except for a few select companies that are the front runner that have been working on this for the last 10 years. So I think at least for us, for Ultragenyx, I think still a lot of kind of efforts needs to be focused on and optimizing the manufacturing process to really enable large scale, consistently making high quality products. So that's one. Okay, maybe I should kind of mention a few kind of technical, on a technical level, few things to watch for. I think some of these has been touched on in previous presentations today. How do you reduce the amount of double-stranded RNA, and how do you make less of the shortmers, and how do you kind of optimize the structure of the LNP to kind of optimize the function or the potency, right? So that's one, optimizing the process. Secondly, reduce cost of goods. Currently, cost of goods is still extremely high. So many efforts can be worked on to kind of reduce the cost, from raw material to manufacturing scale to yield, to purity, all the way down to cold chain. How do you get away from the minus 80 degree storage and shipping and distribution, right? And how do you reduce the number of samples for analytical testing? And that adds to the cost as well. So we're working on some of those. Okay, so other than optimizing the process, cost, the other one is scale up. Scale up is a big issue. I think the current pandemic is really providing a kind of an urgent catalyst to really accelerate the speed from bench scale to large production scale. I think quite a few companies that have done a huge amount of advancement in that area. For us, it's still an area that we would work on. So lastly, I want to mention analytical technology. Lots more analytical characterization or tools needs to be developed for this field to progress, for to mature. Currently, we're still very much relying on using animal models to look for potency. I think one area we wanted to focus on is really develop characterization tools to really establish and understand the relationship between structure and function.
Good. Thank you, Sharon. And again, one follow-up question comes to my mind is, I completely agree, as the technology matures, the manufacturing process matures, there will be more characterization, more, across, as well as, consistency and reduction in cost. But just at a high level, really high level, if you were to compare antibody manufacturing production or viral vector production, those manufacturing lines versus an mRNA LNP line, at this time, would you say that the mRNA LNP manufacturing is simpler, faster, cheaper, or that's not really true?
At least for us, most of the companies we're not there. I think eventually it will be faster and cheaper. The whole making of the mRNA and the lipid nanoparticle, especially on the mRNA side, it's not a cell culture process or in vitro prescription process. So it should be faster and simpler in the future. Right now we're not yet there.
Yeah, okay, makes sense. Robin, anything you'd like to add here with respect to how do we take mRNA LNPs mainstream?
Yeah, I don't want to kind of just repeat what Sharon says. She did a very good job there. So I'll just try and mention a couple of things that I don't think were covered. Obviously the scale out, but that could, you could scale out rather than scale up. I think that's an important consideration. And the other aspect I think at the moment is actually the speed of production is fast, but the bottleneck then becomes the fill finish. Actually, we can produce more vaccine than we can get into, vials, and so that becomes a bottleneck. I don't think anybody at the moment is thinking about how to put mRNA vaccines into pre-filled syringes and keep those in a way that you can ensure a storage. So we need to think about new ways of delivering LNP RNA-based vaccines. And we're definitely not there with cost. If we think about the viral vectors, for example, in COVID-19, the AstraZeneca adenovirus vector requires only refrigerated temperature for 2 years, and it's coming in at a cost of $3 a dose. Whereas the current RNA vaccines are, above $20 a dose. So if and when we see the viral vectors come through, we may see that actually the pressure on getting mRNA vaccines out the door is diminished overnight if there's something that shows the same level of efficacy but is a fraction of the cost and doesn't have the cold chain requirements. So we're sitting in this kind of honeymoon space where it looks like RNA is the only game in town, but I don't think that's going to be the case for too long. And for the future of RNA vaccines, we need to solve cost, stability, distribution in fairly short order.
Perfect. Thank you, Robin. So for my next question, and again, I'll start with Kurt here. In your field using mRNA LNPs, what do you expect to see in the next 5 years? So for Ceva Animal Health, products to be launched, and where do you just see investing and moving forward with products in the field?
Great question. So we are very excited about the opportunities that RNA presents for the veterinary world, specifically in the production animal. So we're very aggressive and we work in very specific species. So swine, poultry, and ruminants. And so our goal in the next 5 years is to have a 1 to 2 fully registered vaccines in swine. The same is true for poultry. I think what is the even more exciting aspect is this platform system. When you get fully registered, at least in the United States and shortly in the EU, you can then use it on a prescription basis. So we could imagine anywhere from 5 to 10 prescription or products in both swine and poultry in that same time frame. So and I think it's going to take a little longer in the EU and outside of the EU because the regulations are still not currently in place, but they will be in 2022. There's a modification of the Annex 2 that for the veterinary use that will allow us to talk about platform systems and the streamlined way in which you can go through the licensing process. As we look at some of the biggest issues, the success of this platform approach for veterinary health will be getting the initial regulatory approvals. And once those are in place, then we'll be able to build rapidly. And so that's why we think we can get so many commercial products out in such a short period of time.
Yeah. And, Kurt, I mean, I'll take the opportunity. There's a quick question, which is more specific for you coming from the audience, which is, are your vaccine targets specific to viral diseases or other infectious diseases? For example, will these products have an impact on the use of prophylactic antibiotic use in animal production?
Very good question. The systems are not limited to viral targets. They can be equally effective against bacterial targets. The key is you have to have a gene of interest, a target that a protective immune response will work against. So as long as we're not talking about a carbohydrate, we're talking about a protein, then the system should work. And the viral particle approach to this self-amplifying RNA has been used in many different arenas: infectious disease, both viral, bacterial, protozoan, and even cancer. So so I think that there's no restriction. As long as there's an effective gene that you can put into the system, then you should have the outcome you want in a vaccine setting.
Perfect. Sharon, maybe you can speak about Ultragenyx. I know you work with multiple technologies, but you are making bigger investments in gene therapy. And so, in the next 5 years, where do you see the area of mRNA LNPs going?
Yeah, sure. We're very excited about the whole mRNA LNP technology and platform for treating rare disease patients. With the amazing efficacy that Moderna achieved this week, and as well as Pfizer BioNTech achieved last week. Basically, for this class of molecule, the proof of concept has already been demonstrated. So for us, we have maybe I should go back to our business model. So as I mentioned, our business model is to bring in the right target, bring in the right technology, and combine that with our deep rare disease development expertise. And this is what our model is. And for that, we currently have a pretty big focus on both gene therapy as well as messenger RNA, as well as oligonucleotides, such as antisense oligos. For mRNA, we currently have a molecule that are heading to IND and clinical development next year. So, with our expertise in rare disease development, we certainly hope to get close to regulatory filing as well as approval for this product in a few years, and along with a few others in the clinic.
Perfect. Thank you, Sharon. And Robin, I know you're out there in phase 1 clinical trials in an mRNA LNP for COVID, but if you can also speak to next 5 years and what you are planning with Vaxequity and really vaccines for the developed world, the developing world, that'll be great.
So we're continuing to work on other pandemic infectious threats, supported by CEPI and other UK funders. So we'll continue to work on those targets. Those aren't necessarily commercial, but they help to drive the technology. We'll be looking at targeting vaccines of unmet need. Obviously, we're a pretty new spin out, so I'm not going to list the commercial targets we're going to go after, but I'm sure people can guess some of the obvious ones. I don't think this technology is going to replace existing vaccines. So things like, rubella, mumps, measles, you're not going to suddenly be able to compete in that space with an mRNA vaccine. So they probably have to be new targets or targets that aren't well supported by the technology that exists. And then outside of the commercial environment, with my kind of academic hat, because of the ease of production, I think that they will be used increasingly in vaccine discovery, where we particularly know that animal models are not always informative. So a lot of my life has been spent in the HIV vaccine space, where we really need to understand the human immune response to HIV immunogens to understand how to induce neutralizing antibodies. And so in the context of vaccine discovery, it allows you to get into early experimental medicine type studies and really drive discovery research in humans rather than the traditional reliance on animal models, which are helpful but not always predictive.
Perfect. Thank you so much, Robin. So this is great because, again, PNI with our technologies and our strategies is planning in the same direction that many of you have talked about. And this is, again, building a library of novel lipids that enable more possibilities across these different therapeutic areas. Very obviously, the manufacturing platform that scales out, scales up, and is able, to manufacture this in the most in the most expeditious, cost-effective fashion anywhere in the world. And then, again, as Sharon, Robin, you have all mentioned, the development expertise, the analytical methods, the formulation, the scale up, and the scale up that is so important. So, we are excited at PNI to be partnering with each one of you in these different areas to move this platform, this technology to mainstream. So I'd like to ask if any of our audience have any questions here for the panel members. And, if Sharon, Kurt, and Robin, if you have anything that you would like to add, this is a great time.
I'd like to add one piece, because I think the model paradigm is an important one. The beauty, and I think what we'll be able to do is leverage the success that we see in veterinary health will help human health, because we don't have to work with models. We can work directly with the species that we actually want to care for. So the safety, the efficacy aspects that come out in this field, I think will help in the long run for messenger RNA or any other RNA approach in human health. And so I hope to play some small role in bringing these technologies mainstream for whatever appropriate targets that get asked production people to work on. So.
Yeah. Sharon, from yourself, additional thoughts, anything you'd like to chime in?
No.
Robin, yourself?
Yeah, I mean, I think it's been an interesting discussion. I think I need to stay in touch with Kurt, because I think driving the cost down is important. And if we think the cost needs to be low for human vaccines, it needs to be really low for animal vaccines. So it'd be really nice to keep that discussion going and hear more about how you think you can drive that cost down, because I think that's going to be beneficial for everybody. And ultimately beneficial for this technology. I see it a bit like the computer industry. The first computers were very expensive, not very accessible. And what really changed everything was when there were many computer manufacturers. and the prices came down and the technology was accessible. And actually that was beneficial for everybody. It caused a lot of commercial success. And at the moment, it's kind of the select few that are able to develop and use this technology. We need to see a sea change in the next 5 years if it's to become a real global player, I believe.
Yeah, no, completely agree. Okay, I'm looking at any questions here. I'll ask this question. I'm not sure. Again, I'll leave it for all three panelists, maybe Robin, more for you, and see if this is applicable, in the work that you do, or, any comments that you might have. Are you aware of the Bill Melinda Gates Foundation providing birth control injection packages for $1 per injection? Any comments? Again, unsure if the mRNA LNP technology and the work applies there, or any thoughts you have. I mean, I know we talked about bringing down costs, for vaccines and for animal, vaccines, humans and animals.
I mean, in terms of birth control, I'm not sure that mRNA is really in that playing field at the moment, and certainly not at $1 a dose. So it's an interesting question, but I'm not sure that we're in a place to address that. And I noticed in the chat, there's a question to Kurt about whether animal models allow you to bridge or fully predict human responses. And I'd say pretty much no. But they're models, they can be useful. And what we've been very conscious of as we get more human clinical data is to work backwards and look and see which models were predictive, how were they predictive, and how can we better use them for, future vaccine approaches. And I think you need to look both forwards and backwards to get them the best out of the animal models that we currently have.
No, thank you. Kurt, we have a question here for you. Can animal model fully predict immunogenicity effects in humans? How to better bridge animal models to human applications?
Well, I think Robin addressed a little bit about that. I mean, there are examples where in say cancer, where work carried out in dogs has correlated to humans, or humans has correlated to treatments in dogs. So there are specific examples where models are helpful. I think my experience with SAM systems has indicated that the dose that's effective, looking from an immunogenicity perspective, the efficacy component I think is an open question, but from a safety or dose perspective, a dose that works in a mouse will work in a guinea pig, will work in a pig, will work in a non-human primate, and may or may not work in people. But so there's less of a dosing question when it comes to SAMs anyway, and effectiveness in the model that you're going after. So yeah, I think the models, as I indicated, is a very important part. And fortunately for me, anyway, working with the animals that actually you want to support, you don't need to worry about that as much. I think it will help though is safety, and is the approach efficacious? Can it be effective? And eventually, will have to be tested in people. So I hope that's some addressing of that question.
Sounds good. Shel, are there any other questions that I'm missing that you would like to pick up here? Shel?
Hey, Padma. There were a couple of questions that just popped up on the Q&A just as you were calling upon me here. So maybe we can try to address those first. Okay. So one question to the panel, how to improve the loaded LNPs to target a specific organ, and what are the challenges and opportunities there?
Maybe Robin, and then Sharon.
I think this is a great question, but it's not something that we have looked at a lot, because in the infectious disease space, we've really been looking at intramuscular administration. And so we're not targeting specific organs other than perhaps, the draining lymph node. But obviously for other indications, it will be very important to improve on the targeting.
Sharon, in the space that you're working on at Ultragenyx? Any interest of extra hepatic delivery?
Yeah, I mean, for now, the more advanced programs that we're working on is targeted on liver disease. We are exploring ways to target other tissues currently.
Thank you. And from a P&I perspective, as we work with 150 plus clients, there is interest in extra hepatic delivery. However, everybody is first looking at the platform with what it can do initially, and then obviously, moving on to, additional targets outside the liver. So at least that's what we hear in our discussions. Shell, anything else I am missing here? Go ahead.
Sorry, I was just gonna also say that, actually, sorry, I've just kind of lost my train of thought there, but there are other questions if you want to move on to a different topic, or do you want to continue on this one?
I think, yeah, we're ready to move on. I think we are at the close of our session. We have just a few more minutes left. Yeah, we're at the close of the session. So Shell, if you want to go to any other topics. First, let me thank our panelists, Sharon, Kurt, Robin, for participating and really talking, on the business side and on the technical side, the programs that you are leading in your respective organizations. Thank you very much.
Thank you.
Shell, to you.
Yeah, so there was a question actually about whether or not the lipid manufacturers or excipient manufacturers are quote unquote ready for the challenge. And I think this maybe points to all of the supply chain potential issues for making LNPs. And where are we with that? And do you guys see this as a potential issue?
And who can, who are you asking, to answer? We do have, Jeff, Peter, Robin, several on the line. Anybody can chime in, please?
So, I mean, I think, are they up for the challenge? Yes. Are they ready to make enough lipid to meet the current demands? I suspect at the moment that we need to see an increase in capacity.
Thank you.
Thank you. So I think we are ready to move to the next session. So Jane?
Yeah. I just have to hold up just a minute because it's actually a third session. So we've got some other questions that came in. If you guys don't mind.
Okay, please go. Yeah,
go start at exactly the 30 at the half of the hour, if we could.
Okay, okay. So Shell, if you could just continue with your questions.
Sure, there were a couple leftover questions from the previous session. If we can pull Jeffrey and Peter back into this conversation, we can have a sort of a larger, broader panel as well. I don't know if Jeffrey, you're on the call there.
Yep, still here.
Okay, excellent. So there was a question about, given the potency of a self-amplifying RNA, is there any advantages for using this in sort of non-vaccine approaches, for example, protein replacement therapy?
Well, I think the two potential attributes of the SAM approach, one has already been discussed, is the dose, lowering the dose, making greater number of doses with the same mRNA, but then the opportunity of making a more potent response is also there, and maybe longer lived expression. So outside of the infectious disease space, for transcript therapy where you want to produce a biologically active protein for a period of time, the comparative studies that have been published and that we did shows that in fact the self-amplifying RNA gives higher peak protein expression and longer lived protein expression than a conventional mRNA. So I think there's some opportunity there to apply this outside of the vaccines space.
Great, thank you.
And we do have a question here for Sharon from Ultragenyx. Since Ultragenyx does not deal with discovery stage R&D, would it be possible to know what business models you establish with the discovery stage academics or startups?
I'm sorry, what's the last part of the question?
The last part is, what type of business models do you establish with the discovery stage academics or startups? So where do you get more products, and I'm paraphrasing this, do you get products which are early in discovery, or do you wait for the products to get to like a phase 1 before you bring them in? Or maybe you do both, don't know.
I see. We're not limiting in that area. We work with we collaborate with academics and other companies for late stage molecules as well as early stage molecules. And in some cases, we in-license the product. In some cases, we acquire the technology and acquire the company. Gene therapy, for example.
Thank you. Shel, over to you.
Sure, thank you. Actually, there was a question that was directed at Peter, but I think the panel also had a similar question addressed to them, which was about the cold storage, cold chain issues with the LNPs. And the question was more about why is this happening? And what is it about the LNP formulation that makes this type of cold storage necessary? And I guess like anyone could answer that question if you have anything to speak to that.
I have a feeling this is a short-lived problem, that simply because these formulations were pushed into production, into clinical trials, et cetera, very, very quickly. It's hard to do a 2-year stability study in a month. So these are issues that I think you'll see solved as we move forward.
Right, and I think Kurt had also mentioned, if you went for more of a just-in-time model, that may also alleviate some of those that necessities.
Right.
Okay, great. With that, we're sort of reaching the bottom of the hour and ready to move on to the next session.